Hello, everyone, and welcome to today's live broadcast, Streamlined Clinical Trials, Applying Rigor in Flow Cytometry to Facilitate Therapeutic Developments, presented by Dr. Alina Lelic, the Field Marketing Manager for Flow Cytometry at Beckman Coulter Life Sciences. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Labroots and sponsored by Beckman Coulter Life Sciences. For more information on our sponsor, please visit them at www.beckman.com. Now let's get started. I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during that presentation. To do so, simply type them into the Ask a Question box and click the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, click on the Support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. Without further ado, I'd like to now introduce our presenter, Dr. Alina Lelic. For a complete biography on our speaker, please visit the Biography tab on the top of your screen. Dr. Lelic, you may now begin your presentation. Welcome.
Thank you, Susie, and thank you everybody for attending this webinar that's going to discuss streamlined clinical trials and specifically how we can apply rigor to flow cytometry to facilitate therapeutic development. The purpose of this presentation and the education itself is really to provide you insight, information, and hopefully some really good practical solutions in terms of your own flow cytometric workflows that are going to progress your scientific endeavors and progress your scientific excellence. So we're going to begin. So how exactly does science translate to medicine? Many of us who are stuck in the lab and we're doing the science at our lab bench, we forget the final outcome and the purpose of why we're doing the science. But ultimately, it's to progress the science into medicine. And today, catching up to coronavirus, which is quite an interesting time that we live in this pandemic, we can see this report that shows 60 treatments that are currently in development. So these treatments obviously came from the preclinical trials that were developed in labs, in animal models. But if we just simply look at the clinicaltrials.gov, what we're going to note is that there are to date 997 COVID-related clinical trials. Of those, 695 are evaluating therapies. So what this shows us is that clinical trials are extremely important in terms of developing the actual methodologies and evaluating the therapies that are in clinical trials. So how do we relate flow cytometry with clinical trials? So in the same token, if we just simply go to clinicaltrials.gov, what we'll note is just if you search into the study title, flow cytometry, what you'll note is 629 clinical trials that are focused around flow. This is definitely an underestimated number because of the fact that I'm looking for the title in the actual search criteria. And there's definitely a large number of trials that are involved in utilizing flow cytometry as a method. So collectively, clinical trial evaluates the medicine, and flow cytometry is the method by which we can actually do this. So how precisely can we utilize flow cytometry in biomedical research? Well, flow cytometry allows us to characterize and evaluate single cells, and specifically how the therapies that we're evaluating modulate the effects on the immune system. So if we're looking at specific therapies in mind, we're going to analyze and assess cells based on their phenotype, function, health, as well as the viability. And in terms of activation and differentiation of these therapies, how they translate specifically on a cell that's cell-based levels to identify potential biomarkers in longitudinal studies and a comprehensive analysis of the immune system. And we do so after the administration of therapies that are being evaluated, such as prophylactic vaccines, drugs that we're looking at, or small or large molecules. So collectively, we have the capacity to characterize the immune response and potentially identify these biomarkers that are going to either allow us to address disease progression or disease halt, so correlates. But what's important to understand is that flow cytometry is, in fact, a quasi-quantitative and a qualitative assay. So it's going to give us numeric and categoric data, which is actually correlative data. So this correlative data, in essence, the method itself lacks calibration standards. We don't have the proportionality to the amount of analyte. And because of this, it is very important that we validate the method for imprecision. What that means is that we need to understand the error in terms of the error of measurement that we are obtaining. And the reason for this is because how do we ensure that this quasi-quantitative and the qualitative assays are reproducible? Because that's essentially what we need to obtain in order to analyze the therapies that are in clinical trials. So scientific rigor is one aspect by which we can obtain reproducibility. But before we delve into scientific rigor, let's specifically define it in the context that we're discussing here. So the rigor is a strict application of our scientific method. What that means is we're going to ensure that our experimental design, methodology, analysis, interpretation, and reporting is unbiased and well controlled so that it can be reproduced by each scientist irrespective, when they do it. So scientific rigor ultimately is going to lead reproducible data for translational purposes that we just discussed of science to medicine, which is going to in turn allow development of immune modulating therapies to treat the patient when it's necessary. So understanding that we have scientific rigor, it's something that's been applied in science for as long as we've been studying it, and considering the fact that the NIH has actually put together a rigor and reproducibility grant that scientists can apply for, why are we discussing this? If we understand it's there, if we know that it can be utilized, why are we talking about rigor? Could it be because there is a reproducibility crisis in science? And unfortunately, that is the case. If we look at the preclinical data that's been generated in the United States, and I'm not showing you costs here, but 50% of the data that is reported is irreproducible. And this wages on various components of reproducibility, which are listed in this schematic here, which you can see are reagents and materials, study design, data analysis reporting, and protocols, all within the control of the scientists themselves. So why is it that these materials and reagents that we can actually control and ensure that we are rigorously implementing into our experiments are producing such a massive and alarming number of irreproducible studies? And ultimately, the irreproducibility leads to delays in therapeutic development and increased costs. And this is something that obviously is unacceptable and needs to be changed. So the reproducibility issues, especially in the bioanalysis, such as flow cytometry, has been addressed in a workshop, an annual workshop that occurs. It's called the Workshop on Recent Issues in Bioanalysis, where leading global biopharma, CROs, biotech companies, as well as regulatory agencies come together and they discuss issues and they develop guidelines and specific information that could be utilized and disseminated across the scientific community to implement some sort of rigor so that we could develop better studies and expedite the development of, accurately expedite development of therapies. And last year, the guidelines for flow cytometric bioanalysis came up with several points that I'm going to address here. The first one was the use of ASR reagents. ASR reagents, also known as analyte-specific reagents. So when it comes to flow cytometry, these reagents will be monoclonal or polyclonal antibodies that are GMP manufactured. And they're analyte specific because of the fact that they've been evaluated for their specificity. They're manufactured in a GMP facility, and they have qualification that comes with the actual product itself that shows you the utility specific certificate of analysis and eliminates you from having to do an in-house evaluation of the product itself. The next aspect of when utilizing flow cytometry reagents is to ensure that you determine the concentration, specificity, clonality, fluorescence to protein ratio, and immunogen information. So when we are dealing with GMP manufactured reagents, such as ASR reagents, this is something that the vendor will provide for you. And it's very useful because, again, it eliminates the need for you to do so. However, if this information, if you are using RUO reagents, it's critical that you actually have a validation and a specification for your reagents when you're doing, prior to you starting your clinical trial. Titration of antibodies. This is something that we hear all the time. It is a critical aspect in flow cytometry because it can definitely produce erroneous data. So ensure that when you do have lot changes of your antibodies ensure that during the duration of your study that you're titrating the antibodies appropriately and that you record these titrations. Inclusion of FMOs for gating. This isn't an issue when we have beautifully separated populations, distinct populations where you can discern positive from a negative. But especially in the case where we have cellular therapies where the antigen of interest is has a modular effect and we can't necessarily discern a positive or a negative, it's important to include those FMOs so that we can utilize them for gating, but not just for gating, but also for compensation matrix validation. Next, it's important to validate and ensure that if you have changed any of the reagents, or if anything any changes have occurred during the duration of your clinical trial, that you validate this. That also means that if reagent lots have changed, you're going to ensure that these reagent lots are validated and that you've cross-checked them against the previous lot. And lastly, when you're reading an MFI, such as in the case of activation markers, CD69, or whatever it is that you're looking at, ensure that you're normalizing your actual instruments with MESF beads so that you can accurately report out the MFI. So now that we've discussed the fact that there is rigor and we have the scientific method that backs it up, and we've also discussed the fact that the scientific community discusses guidelines and workflows, we're going to show you how to implement streamlined workflows into your own cytometric assays. And this is quite simple because based on the chart that I previously showed you, you can see the components of irreproducibility to the actual global problem of reproducibility. And so what we're going to break down the streamlined workflows into is three aspects. The first one is going to be sample preparation. So the important aspect of sample preparation that we're going to hone into is limiting reagent variability. Next aspect is minimizing or automating pipetting. And lastly, or if possible, automate the sample preparation itself. So collectively, by sample preparation, we're going to introduce a streamlined workflow that's going to be reproducible. The second aspect is data acquisition. This comes to your instrumentation. What kind of flow cytometer are you using? Have you calibrated and evaluated your instrument performance and stability? And have you standardized your instrument setting during the longitudinal study that you're evaluating? And lastly is the data analysis. A lot of inconsistencies come from manual gating. So what we're going to try to do is highlight ways that you can actually limit these inconsistencies and utilize unbiased platforms to analyze your data so that you can reproduce it and not have to delay. So the reagents, when we talk about the reagents for the clinical trials and experiments that we're looking at, we've discussed the fact that the ASR specifications are going to give us qualifications and QC report out. So ultimately, what we want to do is limit the validation that is needed in-house, because that takes time, that takes effort, and that can introduce additional errors. So what we're going to do is we're going to leave that to the vendor. We're going to obtain unique reagent identifiers from the vendors, and these could be anything such as ASR specifications, certificate of analysis that come with reagents, GMP or ISO specifications. So some reagents may be ISO specified. They have their own pre-qualified QC procedures, but they're not necessarily GMP. So these are all very useful specifications and unique identifiers that you can actually obtain from the vendor. Next, we're going to minimize reagent lot changes, and actually we're going to minimize changing any reagents during the duration of the study. So prior to you developing your panel design, make sure you validate and evaluate the panel so that you can lock it in and ask the vendor to lock in the lot. Now, obviously, and most likely, you won't be able to lock the lot for the duration of the entire study, so let's say 3 to 5 years. But what you want to do is to limit the number of lot changes, and this can be easily done. The next aspect that we need to ensure is that the reagent stability is evaluated. Again, this can come from the vendor itself, and a lot of the reagents that come with specifications will have an expiry date, so that takes away from you having to do it in-house. Alternatively, you'll have to check for the stability. What that means is check for the expiry, check that the fluorescence hasn't diminished over time, and check it to ensure that while you're actually, cocktailing your antibodies, you're not decreasing the actual fluorescence, and that's not causing a degradation of your tandems. And lastly, and most importantly, we're always going to incorporate experimental control within our daily workflows. This experimental control is the QC to our entire workflow, much like the QC of the instrument and the QC of the reagent that comes with specifications. So we're going to discuss this in a little more detail. Now, I just want to show you for illustrative purposes the reagent documentation that come with some of these ASR reagents specifically. So they'll come with their own pre-qualified specifications, and these specifications will show you the clonality, as well as protein to fluorescence to protein ratios. And in terms of the certificate of analysis, you'll get the expiry date, as well as the batch record verification, which tells you that the reagent itself conforms to the QC specification. In addition, apologize for that. In addition, you'll get a certificate of analysis that is a little more detailed, that's going to show you specifically what results are being passed, so that you can understand the type of QC that's involved in your reagent. Now, if we discuss incorporation of experimental control, most of you will say, well, some of you will know what it is, but what exactly is an experimental control? So what this is, it's an internally qualified sample that contains your antigens of interest. So what I mean by that is, let's say you're reporting out your CD3, CD8, like your subset ratios or your subset percentages. What you're going to do is you're going to acquire a leukapheresis product and you're going to freeze it down into aliquots. And every time you run your experiment, your internal control that you've already validated is going to be incorporated into that experiment. And when I say validated, what I mean by that is that the actual product, or your leukapheresis sample, is going to be run 20 times individually prior to you setting it up into the experiment, such that you can assess the ranges of percentages of interest for your antigen of interest and the actual ranges that are acceptable. So what we're going to do is we're going to basically generate an assay sheet that is acceptable for your own experimental QC. Now, there are manufacturer standards that are available for purchase, and we all know that they exist: Cytotrol, Immunotrol, and various other cell types that are either cryopreserved, preserved utilizing specific chemicals, or lyophilized. So you can use these for your daily verification of agents, and ultimately, you always want to include in experimental controls, because not only is it going to give you the QC of your experiment, but it's going to tell you whether your instrument is stable, whether you've incurred any pipetting errors when it comes to cocktailing your antibodies, or if something has just gone wrong with the experiment itself. So the schematic here shows you a cytotrol sample here that comes with an expected values and ranges. that shows you the percent positive. So let's look at the CD3, which is the second or third row from the top. And CD3 percentages are 78% plus or minus 8. If we look at the second plot, flow plot, you can see that the CD3 percentage here is 73, within the acceptable range. So once you run this sample within your experiment that you are running on a daily during the longitudinal study, you're basically just going to set it as a pass and incorporate this into the QC of your experiment. Quite simple. Now, we discussed the fact that we can introduce error into our cocktailing, and we discussed the fact that this is another reason why we're going to have an internal control or an experimental control. So the liquid cocktail antibody looks like this. We first design our panel. We select the antibody clones, we select the dye combination based on the antigen level, based on the fluorescent intensity, and we evaluate and verify the panel performance. There's going to be several iterations of the verification of your panel performance because that takes some time. And the more fluorescence or the more antigens we want to analyze, the more laborious this process becomes. But ultimately, once we verify the panel performance, we're going to say that it's okay. We're going to optimize the preparation protocol, which comes in hand with the sample preparation and everything that entails into that methodology. And we're going to create an SOP for cocktailing, mixing, as well as the QC. Once this is all done, the assay itself is transferred into the lab. So ultimately, there's several checkpoints where we can actually introduce errors, cocktailing errors, panel design errors, even in the case of creating an SOP and making the cocktails. There's several ways that we can introduce the error. So if we look at the reagent variability within the workflows, you all know that staining patterns and intensity really depend on pipetting accuracy and reagent shelf stability. So if a tandem dye degradates, you're going to get a diminished signal that you're not necessarily going to know that it's related to your reagent. You may think it's a biologically relevant aspect, but it's not. So it's important to understand that antibody titrations, cocktailing, and antibody validations themselves are critical aspects of making sure that your workflows are rigorous. But they do introduce risk of variability and error if you have to consistently do them. Not only do they increase the error, but they also lead to loss of time and loss of money, which is something that obviously we want to make efficient. So the question begs, do we have an alternative method to this laborious workflow that is currently available? And it turns out that there are. There are various vendors out there that have optimized the cocktailing workflows by introducing dry and optimized ready-to-use cocktails. And so these cocktails are basically dried down antibody panels that have been pre-designed by experts in the field that have been validated and QC checked for you. And so they eliminate that entire section of panel design. And the only aspect of that you need to, so first and foremost, they're dried, so there's no cocktailing. So the error of pipetting goes out of hand, or you're limiting the error of pipetting because you still have to introduce the sample into the tube. And so what you do by utilizing these pre-designed panels and dry panels, you're going to verify the panel performance, test the lot, and transfer the assay into the lab. If it checks in, that it's great. So ultimately, what we're trying to do by utilizing these dry and optimized cocktails is really increase the efficiency of our workflows so that we can, in fact, progress the therapeutic development. And ultimately, what we want to do is ensure that these panels allow us the reproducibility that we were lacking before due to the introduction of errors into the workflow. So ultimately, Beckman Coulter does have these panels, and I just wanted to highlight the fact that these panels were actually something that the key expert opinions that were involved in the one study, this was a transplant experiment that was done across the globe, came to the actual scientists and said, we need stabilized panels because reagent consistency and reliability is something that is questionable. So how can we introduce these panels that we can ship to Europe, North America, and ensure that that there's no pipetting errors, so that what we're looking at is truly biological relevance. So the scientists at Beckman Coulter got together with these key expert opinions, and they developed 8 immune phenotyping panels that are listed here. And basically, this collaborative effort led to a beautiful design of these 8 panels that can characterize the entire phenotype of the immune system, technically. And what I wanted to show you here is a couple of examples of these tubes. Now, it's important also to qualify that a lot of these panels are going to bear their own quality standards. This one specifically is under a specific ISO manufacturing standard, which says that it comes, which states that it comes with its own QC specifications. Now, if we look at the actual fluorescence, if we look at the panels here for the IM-Treg tube, what you'll find is 8 antibody conjugates that define and differentiate your Treg populations based on FOXP3, CD39, and Helios. What's important to note is that there are 8 antibodies, but based on the configuration of your own cytometer, you can actually add additional antibodies if you want to characterize your Tregs further. So let's say you wanted to look at the memory marker. You can introduce CD45RA, RO, CD4 is already in there, but you can introduce CCR7 and additional markers that definitely can be dropped into the panel. With respect to the IM granulocyte tube, we're looking at the phenotype of your granulocytes based on CD15 and CD16, but additionally, you have the capacity to do additional drop-in antibodies if you want to take the characterization further. Now, the last tube that I'm showing you here is the IM count tube, and this tube is extremely useful for individuals that are doing drug discovery because all it contains is the CD45 counting bead 7AD, and you can easily drop a PI into the actual mix and look at the proportion of your healthy cells based on apoptosis, necrosis, and various other markers, really. Now, in addition to the phenotyping panels, functional characterization of the immune system is of great importance. And when you are looking at clinical trials, evaluating the functionality of your cells is something that is also critical. These panels, again, provide robust cocktails that can be reproducibly used across multi-sites. And so utilizing them and taking out the pipetting errors of cocktailing and so on, you can look at the activation of T cells, T helper cells, monocytes, and basophils. And ultimately showing you here a sample that differentiates the functionality of your CD4 T cells based on IL-4, gamma, and IL-2 and IL-17. So ultimately, you can still see that there are various drop-ins available here. So if these markers are not enough, and if you want to look at additional activation markers of your T cells, you can do so by dropping them in. So the panel itself serves as a base or serves as the backbone that can then have additional antibodies for further characterization. And lastly, if for any individuals who are doing clinical trials and to detect rare events, there are several panels that are commercially available, and these can assess CLL, multiple myeloma, as well as ALL, and they contain various markers here that I identified. So based on the ALB tube, in the case of clinical trials for ALL, we're looking at rare events that are CD10 positive, and you can see the speckles of those rare events that have been obviously emphasized by the large dots, but you can see the differentiation of the rare events in comparison to the normal. So collectively, these panels have been validated and have been, they bear a quality standard with them, but ultimately they allow individuals who are doing multi-site clinical trials to really implement rigor into their workflows. And in addition to that, they reduce reagent variability. They're stability tested antibody panels, so if we compare them to the liquid reagents, you can clearly see that the comparison between them is on par. The brightness is preserved, there's no changes in compensation. So the ease of use does not come at the actual expense of losing the brightness. So this is extremely important. In addition to that, these panels from most vendors are room temperature stable, which is wonderful because they don't require refrigeration. So they're also stable at extreme temperatures. And this is just to show that, utilizing these panels is really a great utility for, let's say that you're shipping them across the world, and if something happens, all you need to do is validate it with a different sample set that you have and ensure that the panel still contains the same brightness across all dyes. So what this goes to show is that there are workflows that you can implement into your own laboratory and into your own clinical trials that are going to for sure reduce the error that's associated with reagent workflows, or reagent errors. So in essence, we can still reduce experimental variability by utilizing automated workflows. And this comes, so what I'm showing you here is a figure of an integrated automation handler with a flow cytometer. And so what this is going to, this automated workflow is going to basically prepare the sample for you, cocktail the antibodies, introduce the staining of the antibodies, spin the antibodies utilizing the centrifuge that's on deck, and then ultimately take the sample that is stained into the flow cytometer, and then if you have integrated it into the LIS system, you can actually export your samples for the analysis. So this integrated workflow is a really the comprehensive solution of streamlined workflows, and I'm just going to show you a video here that highlights how specifically it works in this particular configuration. Okay. So now that we've seen how the automation works, we're going to discuss what kind of sample output do we get from it. So in this particular case, we wanted to reduce the experimental variability utilizing the automated workflows. And we did so by analyzing 96 well replicates in precisely what you saw in the video there to assess the phenotype of T cells and really evaluate the intra-assay reproducibility. So what you see on the left-hand axis is the mean of the phenotype in terms of the percentages of the cells that are described on the bottom axis. And what you see on the right-hand side is the percent CVs. So collectively, the analysis of the T cell phenotype showed less than 20% variability among the 96 well replicates. And that's actually quite great, especially when it comes to the fact that even in the cases where the percentages of the positive populations were 2% or less, where the variability is going to be the most based on the fact that it's the lowest numbers. So what we get here is really the capacity to reproduce our intra-assay and ensure that our sample workflows are indeed reproducible. So now that we've talked about the reagent rigor and how we introduce reproducibility with our reagent workflows, we're going to actually move on to the data acquisition. And data acquisition is going to discuss the instrumentation. So flow cytometers are inherently different. We know this based on the optics, fluidics, detectors, and electronics. And ultimately, today, you have the capacity to choose any flow cytometer. And if you do so, the most important aspect is that every instrument is evaluated and qualified prior to your use. Ensure that the IQ/OQ has been provided by the vendor and that the instrument has been validated in terms of the functionality. But then, in addition to the IQ/OQ, it's critical that you evaluate your instrument based on quantum efficiency, electronic and optical background, as well as the spillover matrix. Why? Because this is going to allow you to see the best resolution of your signal and to ensure that the signal that you're detecting isn't noise, but it's actual true value. And why, again, is also to introduce this reproducibility that we're going to discuss in terms of the data acquisition. Now, also, understanding the spillover spreading matrix. If you are generating your own panels, you need to understand the detectors and how different the spectral overlap of the different detectors. So this is something that you will definitely need to evaluate if you are designing your own panels. In addition to that, it is important that if you are doing clinical trials and you're going to be reporting your data to the FDA, utilize this 21 CFR Part 11 compliance tools. These are the FDA mandated tools that basically state that electronic records and signatures are considered trustworthy, reliable, and can be equivalent to the paper records that you have. So many instruments do have the CFR Part 11 compliance tools. Utilize them accordingly and make sure that you sign off and that all the data is tracked. In addition to what we talked about in terms of calibrating your instrument, it's important to optimize your instrument settings. What does this mean? It means that you inspect and you understand the entire dynamic range of your instrument. You can easily do so by simply running your 8-peak beads and understanding the resolution of the fluorescent signals. Now you know that it depends on instrument sensitivity and the overall performance. So what that tells you technically is where exactly your instrument sensitivity is and how far you can push your analysis, where you can place the, which channels you can place your dim markers on, and how you can analyze your samples the best. The additional aspect of instrument setting optimization is going to enable at best resolution and ultimately consistent data acquisition that then translate into the next aspect, which is the standardization of instrument settings. This is very critical. If we are looking at longitudinal studies, it is critical that we standardize the optimized settings that we've evaluated utilizing a bead particle. What this means is that we're going to use the same standardized setting for the duration of the clinical study. And the purpose of this is to, with assurance, state that biological differences that we're seeing from a sample from day 0 to day 30 or a year out is actually due to the biological difference and not the setting shift. So what we do is we utilize an actual bead particle and we set the target channels so that these target channels are acquired on a daily. The standardization as well as the QC protocol, much as the inclusion of the experimental control, must be done on a daily and they must be tracked. So the reason for this is because the standardized instrument settings enable reproducible data acquisition. And this, again, is important for us because we want to ensure robust development of methods, especially when it comes to flow cytometry, so that we can progress the therapeutic developments and bring them into medicine. Now, it isn't enough to just optimize your setting and to standardize them. It is critical that you verify that these settings are actually accurate, and not just the settings, but the compensation matrix. So what I'm going to show you here is just a simple subset panel that was done on PBMCs. And what you're looking at here is the live dead discriminator in the zombie red channel and CD3, and then gating on CD8 and CD4. So what you see here is in the first sample, 99% of your actual PBMCs are CD3 positive. So that comes a little bit of a lot. This comes as a bit of an alarm because most of you will understand that 99% is a bit, it's a bit high. So then you go on to validate, and if you look at your compensation matrix, you can see that some of these values are really high. And you may think to check, based on your FMOs, the compensation matrix. So after you check your compensation matrix and you adjust the compensation matrix, you realize that the actual population of CD3 positive events is 79%. And you realize that the compensation error occurred due to the overcompensation of your zombie red that spilled into your BV605 channel. So, oops, sorry about that. So what this says, if you were reporting out your CD3 values, you would basically report out erroneous data. And that would be a problem because in the end, unoptimized acquisition setting or compensation would actually generate this erroneous data, which wasn't accurate. And if you were to try to reproduce this again, utilizing different settings that were optimized, you would not get the same outcome. So this is definitely a problem that we need to watch out for. Now, lastly, we're going to discuss data analysis, which again becomes extremely cumbersome because if we look at, as we increase the complexity of our data analysis and increase the number of parameters that we're actually analyzing, we realize that there's challenges of high dimensionality data and analysis, and not just from analyzing these few flow plots, but also as we increase that number, it becomes extremely difficult to analyze that many plots. And ultimately, there is a bias that's associated, but more so variability in manual gating. You're not going to consistently introduce the same gating methodology from one sample to the next. And ultimately, there's, as I said, user bias in gating. What I consider a positive event, you may not. It's going to be easy when we have these beautifully discerned positives and negatives, but when there is a modular antigen in hand or in question, things are going to differ. So how do we ensure that we can actually reproduce data analysis results? Well, and it becomes a challenge. So what if I told you that we could actually reduce the number of plots and eliminate the bias? And it's quite simple. There's algorithms that can do so. And so what if I told you that all of these plots that you're seeing here, we can just simply convert into this beautiful one Visney plot that separates our populations based on these islands and segregates them out based on likelihood. So if you look at all of these events in these islands, they segregate out based on events that are highly similar together. So the Z value here is a CD8. So based on the heat map here, the island that shows CD8 or where your CD8 T cells lie is right there. So we've basically taken out that complex analysis of multitude of biplots and translated to a simple one that we can then further analyze and segregate into metaclusters and differentiate the expression of several different markers utilizing a flow cytometry analysis algorithm. So in essence, unsupervised and supervised machine learning algorithms that are readily available that spans from SPADE, Citrus, Visney, Flow Cytometry, these are just a few that I'm outlining here, that literally have the capacity to reduce dimensionality like we saw there, cluster, identify differences between our data sets. So if we are comparing one treatment to a different treatment, we can clearly see differences that come without the bias of gating that we initially saw from the biplots. Or we could utilize clustering map and identify basically populations of interest that can then be even further segregated down to understand relative expression levels. So ultimately, the utility of unsupervised and supervised machine learning algorithms reduces the complexity of the data without actually losing the complexity of the data. But it eliminates the bias of the manual gating, which is extremely important when we're doing the variability, when we're dealing with variability of clinical samples. And any of you who have dealt with clinical samples, you'll understand that there is a massive variability from sample to sample. And then ultimately, we objectively want to discover new populations, whether these new populations lead to biomarkers or whether they give us prognosis factors. This is the ultimate goal, so that we can understand how the therapeutic development is actually progressing. But ultimately, what we need to do is apply the rigor in reagents, analysis, as well as acquisition to ensure that this is the case. So ultimately, what is the significance of rigor in clinical research? It is to implement efficient technical transfer whenever we're dealing with multi-site clinical trials, longitudinal analysis, and so this is important so that we streamline and develop streamlined workflows to reduce error and reduce variability so that we can repeat these assays and ensure that what we've reported in the preclinical study is as accurate as what we're reporting in phase 1 through whatever. What we want to do ultimately is reduce the experimental costs. These trials cost a lot of money, and we want to reduce this cost so that we can expedite the process. The rigor in clinical trial is going to allow us to generate efficient and reproducible workflows. And the ultimate goal is to utilize unbiased evaluation of therapies in clinical trials so that we can progress these therapies into medicine. So in the end, reproducibility through rigorous scientific method validates what we've seen previously in our preclinical data and allows for progression and the development of immune therapies that are extremely important to the humankind. So I'm just giving you a few resources here. that if any of you are interested, you can click on these links here or ask the questions. But ultimately, I want to thank you for your time, and I hope that you have lots of questions for me. Thank you.
And thank you, Dr. Lelic, for that informative presentation. And we will now start the Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Simply click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. Our first question coming in, Dr. Lelic, is if you do not have access to an internal control cell, is it appropriate to use beads?
Okay, so we're talking about an internal control. No, and the reason, I mean, your beads you're going to use for your standardized experiments, but what you want to do is ultimately utilize the actual a biological sample that you're going to gate utilizing a specific gating strategy so that you can see the progression of the gating and so that you can see if there's any drifts or shifts in the fluorescence intensity. So beads might shift slightly, but you won't be able to see that actual biological relevance onto the population. So an internal sample itself, the bead itself is going to be utilized for your QC of the sample, but with respect to utilizing it as an internal control, as an experimental control, it isn't sufficient, unfortunately.
And does the variation come from antibody design or probe conjunction, and is there a choice of the probe?
A choice of the probe. If you don't mind just elaborating on that. But yes, the variation can definitely come from antibody, and I'm assuming you're asking about the panel design. So the panel design definitely can have some variability. This is why we're going to include our FMO, and we're going to test the antibody panel with single. So let's say we're designing a panel. What you're going to do is you're going to include just a minimum number of antibody conjugates to understand the actual true percentage of the population of interest. And then you're going to start introducing additional antibodies into that. Once you start taking out your fluorescence -1, you're going to see if the actual introduction of new antibody conjugates is going to play an effect on the actual percentages that you're reading out. So this would be the accurate way of designing your actual panel and validating it.
Thank you for that. And I want to thank our audience again for the great questions coming in and remind you that any questions that are not answered today will be answered via email. Our next question also has to do with design. When designing and planning a project with dried down, custom designed multicolor panels, would you suggest testing the panel with a wet antibody cocktail to ensure the performance is correct?
You can, but ultimately what you're going to do is you're going to reach out to the vendor, and they have their own application scientists who understand their reagents, understand the clones, and they have the capacity to design these panels for you. So what you can do is work with the application scientists who will ensure that these panels are probably, that are going to give you the best emission spectrum. However, if you do want to evaluate, let's say, this is a brand new panel that you're analyzing and you've designed it together, we're most likely going to test it in-house. The manufacturer is going to test to ensure that you're getting, and they'll give you the antibodies most likely to test to see if the percentages of the report outs are accurate, and so such that the panel that you're designing together gives you the best readout.
Thank you for that. Our next audience member says, great presentation, thank you so much. And for the dried down antibody multicolor panels, can you add an appropriate fixable live dead marker? And his second part of the question is, can you add in BV, BUV, or BB conjugated antibodies to the panel?
So at the moment, Beckman Coulter does not have, we cannot dry down brilliant dyes, Sturgeon dyes, and the reason for that is they are extremely sticky. So what happens is when you add them, when you dry them down, they lose the actual intensity because they stick together. So we don't have the capacity. I don't know with other manufacturers, but based on the actual chemical compositions of dyes, I'm tempted to say that this would be not an easy process. So the BBs are not easy to dry down, but it's being worked on. And the second question, the first question, sorry, that was, do you mind repeating it again? Sorry, Sergi?
Sure, no problem. It says, given, oh, I'm sorry, I think I've lost that part. Hold on one second, I apologize.
My fault.
I apologize. I don't think I can repeat that part. Let me see if we can bring that back up. And while we're doing that, let me ask you the next question.
Thank you. Sorry about that.
No worries. Given the option, would you recommend stacking as many antibodies as possible in one tube, or splitting it into multiple tubes? And for the second approach, how would you combine population frequencies reported in different tubes, given that there is not a universal parent?
Okay. So if we are stacking antibodies, can definitely be applied to your panel. If you're doing algorithms, if you're going to analyze your data utilizing algorithms, there's going to be a little more manipulation in terms of the manual gating that you'll have to do to begin with. So me personally, I prefer to separate my tubes and then look at the analysis rather than stacking them. But ultimately, you can stack and do some pre-qualification of the actual data, so manual gating, and then importing those specific gates to look at the analysis of the populations of interest. Now, in terms of how do you amalgamate the data, if you are using, you can merge data from different tubes. Now, I know that Kaluza analysis allows you to do this. And so basically by merging the data sets, if it's the same tube, it would have to be the same panel, unfortunately. So you can do this in the algorithms. So there is a capacity by which, if it's the same sample, obviously, and you have different sample sets, there is a way that you can actually merge the data sets and look at the expression of the antigens.
Thank you for that. And which software would you advise for complex data analysis?
Well, that's a really good question, but it depends on what you're trying to assess. So, I mean, the great thing about all of these algorithms is you can layer them. So, for example, most individuals who are doing complex data analysis, they're gonna start with VISNI, and then they usually layer FlowSum onto it. But if you are working with, let's say, patient samples, Citrus is a really beautiful algorithm that allows you to look at, you know, biomarker identification and differences in the treatment options and so on. So ultimately, you have a various platform of different algorithms that you can utilize. The best thing to do would be to actually reach out to the actual data scientists or the application scientists for the different softwares and ask them. Or alternatively, try the different ones and see what the data gives you.
Thank you for that, Dr. Lelic. And thank you again, audience members, for these great questions coming in. Our next question, in discussing the dry optimized antibody cocktails, are these the only options available in terms of antibody antigen combinations, or can different panels be designed?
Yeah, so these are just the ones that are available off the shelf. But I'm sure that, well, I know Beckman Coulter for sure has a custom design services where you can sit down with an application scientist and an expert and design your own panel of choice and evaluate that panel and then transfer it to your own laboratory. So they're not the only ones.
Thank you. And what is better getting control, an FMO or internal negative control?
So if you have an internal negative control, that's going to be the best choice for your, because then it precludes you from wasting additional reagents on including an FMO. However, so FMOs can be used if you are using PBMCs or whole blood, because you have a slew of populations that are not expressing your choice, your fluorescence marker or your antigen of choice. So you have the capacity to do so. However, for clinical trials that are evaluating cellular therapies that have been that have undergone that have basically gone through the process of expansion, and you have one population of interest, or you have somewhat a homogeneous population, in that particular case, FMOs are the only option you have.
Thank you for that. And we have time for a couple more questions. This next question is a two-part question. Can you explain how you're using TSNE to bypass compensation matrices? And it was not clear to me how you use uncompensated data to segregate populations. How can you differentiate true signals from noise?
So you should never use uncompensated data. And I'm really sorry that if that was lost in my message. It wasn't what I was trying to say. So you should always compensate your data, and the data that I was showing you in this slide here, this data was actually compensated. Are they able to see my slides, or are the slides gone? I'm just curious.
Yes, you can still see the slides.
Okay, perfect. So the data example that I'm showing you here, this was actually compensated data. And how we evaluated that the compensation itself was inaccurate was by checking single stain controls with the cells and evaluating to see whether or not really the CD3 population was accurate. Once we established the accurate numbers, we realized that there was an overcompensation, and we manually changed the compensation. However, for future analysis for this particular experiment, this wasn't an experiment that I did personally. This was a sample set that was given to me by one of our colleagues. So what I would do for the future of this experiment, I would actually ensure that the settings as well as the compensation matrix are optimized by utilizing these single cell controls to ensure that's the case. And then there was a second part to the question?
Yes, how you use, well, I think that was the second part actually. Can you explain how you're using TSNE to bypass compensation matrices?
TSNE, TSNE, I believe. Okay, so TSNE so you cannot utilize TSNE to bypass compensation. If you bring in uncompensated data into your TSNE, it will not work. So what you need to ensure is that prior to bringing your data into the algorithm and importing it onto the Cytobank platform, you have to ensure that the data has been QC'd. So what you'll do is you might use a software like Palooza or Flojo, and you're going to ensure that the compensation is accurate by, again, looking at your single cell controls and then ensuring that the compensation is accounted for. If not, then check for it, adjust it manually, obviously, but by using the FMOs, and then export your data into Cytobank for analysis by TSNE.
Thank you for that. And you showed several dry cocktails that are available by Beckman Coulter. Are these specific for Beckman Coulter's instrumentation, or can they be run or other flow cytometers?
No, so they're not specific to Beckman Coulter instrumentation. The beautiful part about these, so these can be run on any cytometer, any commercially available cytometer that has the appropriate bandpass filter, obviously. So we provide you with the dye, and you can see the emission of it in the IFU for the product itself, and then you can basically run it on any platform.
Thank you. And Dr. Lelic, can a lab use the internal control to evaluate getting strategy and applying the control gating strategy to the clinical samples?
So if the internal control is if the internal control mimics what you have as your sample, as your clinical sample, so let's say it's a PBMC, you can kind of gauge it, but I would advise against it. And the reason why is because we talked about, I touched upon sample variability. All samples are different. So in terms of, looking at separation of antigens that are, very discrete, sure. But if you are looking at modulating effects, modular expression of markers, in that particular case, your controls are going to be needed. And those controls are either, your day 0 of treatment and versus your day 30 or your FMO. So those are going to be needed.
Dr. Lelic, thank you for your presentation today. And do you have any final comments for our audience members before we close?
Stay healthy and stay safe. And it's been a real pleasure. If you guys have any questions, please reach out to us. We'll be happy to assist you in making your science excellent. That's the goal for what we're trying to do here.
Dr. Lelic, thank you again for your presentation and for your important research. And before we go, I want to thank audience members for joining us today and for their interesting questions. And questions we didn't have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of our registration. We would like to thank you again, and we also like to thank Labroots and our sponsor, Beckman Coulter Life Sciences and Labroots, for underwriting today's educational webcast. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, bye-bye and stay healthy.